Welcome back to Dornsife Dialogues. It's a pleasure to have you join us for our first event of 2022. We begin this year with an all too familiar question. Will we ever get past this COVID-19 pandemic? The one thing we know for sure is that the expertise of our medical professionals has been and will continue to be invaluable. Our terrific panel of experts from USC will provide insights today on how we might expect COVID-19 to evolve in the weeks and months ahead. They will also talk about current efforts across the scientific community to understand the possible lasting impacts of the pandemic. And they will allow plenty of time to address your specific questions and concerns. Moderating the discussion is Peter Kuhn, who serves as Dornsife Dean's Professor of Biological Sciences. Professor Kuhn's research focuses on personalized medicine and patient care. He is also a key member of the team that identified the most likely order in which COVID-19 symptoms first appear. We're also joined by Paula, Distinguished Professor of Molecular Microbiology and Immunology at the Keck School of Medicine. Professor Cannon is frequently called upon by the media to provide expertise on the ways COVID can be transmitted. She is Associate Director of Cross-School Programs for the MESH Academy and serves as a special advisor to the USC Provost. Finally, Dr. Sarah Van Orman is Clinical Professor of Family Medicine and Associate Vice Provost for Student Affairs. Dr. Van Orman provides leadership on campus public health responses to the pandemic and oversees medical, mental health, prevention, and education initiatives. I cannot overstate the importance of her leadership throughout the pandemic here at USC. There's a lot to cover today, so I'm going to turn the program over now to Professor Kuhn and our panel. Thank you all for joining us, and I wish you a happy, healthy 2022.
Great. Thank you, Amber, and thank you, Sarah, for the introduction and setting this up. It is, of course, a great privilege to be moderating and having this dialogue with two dear friends, Paula and Sarah, who actually I've never met in the real world. So I guess our relationship is a direct result of the after SARS-CoV-2 outbreak. And again, the topic doesn't really require much introduction because we have all been affected by it. But when we were asked in October about orchestrating or putting together this dialogue, it all seemed pretty interesting. The numbers were going nicely down, the daily infections were going down, the daily deaths were going down. It was all going the right direction. And the three of us, like so many in our community, were asking, like, I wonder what the winter surge is going to be like. We saw outbreaks around the world. Everybody was grappling with Delta and what the Delta variant, how that's going to play itself out. But now, of course, through 2 weeks in December, the Omicron outbreak started happening with numbers going up over a 2-week period by tenfold in many places around the world. So we really have to think our way through this, both the virus and how the virus evolves, how the disease changes over time with the different variants of this virus, and how we deal with that both in this country and really around the world. Of course, in the US, by now we have had over 70 million registered infections. We have had over 850,000 people die from this disease. But on the upside, we have also administered over 500 million doses of the vaccines just in the US alone, which is, of course, which is fascinating because together, the vaccination together with the other measures, we have made a big difference in how we handle the virus and how we deal with this outbreak overall, how we can actually protect ourselves and each other across the community. But with that, of course, comes a lot of additional work as we go forward. The good news is, of course, as everybody has been seeing, that the Omicron outbreak is driving cases high, very high, sky high. But the hospitalization rates are not seem to be tracking at the same pace. And there's a lot of questions around that. Because, of course, if we look around the country, we still see a lot of hospitalization utilization getting maxed out. So there's an interesting balance. There's lots of complicated questions. But so let's come to today's discussion. And we call this the evolution of COVID-19. And that was quite purposeful because we didn't just want to focus on the evolution of the virus, the SARS coronavirus 2 virus, but we really wanted to have a conversation around How does this disease affect our society, us as individuals? How has the disease been changing over time? And then with that, this other nagging question around what are the long-term implications for people who have had COVID-19? Of course, this is a USC event. So the three of us will chat about this, but we will actually take a bit of a USC lens at this topic to really give you a somewhat distinct, very consistent with the overall picture of the science and the clinical picture, but with a bit of that USC lens, because we do think that is what becomes most meaningful, because we are a very large community, of course, of students and their families, our alumni, the professional staff, the faculty, as well as our neighborhoods really around Los Angeles. So with that, I will hand it over. To my right is Paula Cannon to both introduce yourself a little bit, give us a feel for your own work in this space, how we kicked it off at USC as this outbreak started happening, and how we got to where we are today. Paula?
Okay, thanks, Peter. Hi, I'm Paula Cannon. I'm a professor at the Keck School of Medicine, and I'm a virologist. My day job is I usually study HIV, but I also study pretty much any RNA virus that comes along. So of course, when SARS-CoV-2 appeared, I was instantly and regrettably interested in it. I've been following this virus now for 2 years and 18 days. And I'm going to give a shout out to UCLA at this point, because it was first brought to my attention on New Year's Eve 2019 by Grace Aldravandi, she's the chief of the Division of Infectious Diseases at Mattel's Children's Hospital over at UCLA. And I think she put it on Facebook or a tweet. She's like, hey, look at this virus that they're seeing in China. And I'm like, oh, is that like at a wet market? And she's like, yeah. And I'm like, ha ha ha, here we go again, Wow. So virologists have a certain amount of black humor, although I'm going to try and not do that in the future. So why were we interested even 2 years ago when we first heard these reports? And it's probably a lot to do with the fact that in the family of viruses that SARS-CoV-2 belongs to, the coronaviruses, we've previously had a couple of really scary brushes with coronaviruses in the world. The virus called SARS, and then a related virus called MERS. And they emerged, they were incredibly deadly. Luckily, they ended up only infecting a limited number of people. I think with SARS, it was maybe 7,000 people, something like that. But because these viruses were so deadly and it was just so obvious you were infected, and because you were kind of only infectious when you had really bad symptoms, it was relatively easy to kind of put draconian measures in place and to really sort of clamp down and stop the virus spreading. So when SARS-CoV-2, it's related, and that's where the SARS bit comes from, came along, I honestly thought we were going to see the same thing, that if this virus started spreading and if it was really causing serious disease, we'd probably go into a period of, lockdown and all these measures to stop spread, but then the world would dodge yet another bullet. And of course, we all know that's not what happened. And there's probably multiple reasons for that. But I think, as everybody probably knows by now, one of the biggest reasons is that, this virus can still spread before you have symptoms, or even if you don't get any symptoms. So it's much more difficult to identify people who are infected and isolate them. So I think the other thing I'll say as a virologist, Peter, is that this family of viruses, the so-called coronaviruses, normally we don't worry too much about them evolving or changing or mutating. They're pretty good at kind of keeping themselves intact, not like viruses like influenza, for example, which mutate every year and we just can't keep up with them. So I think early on, we all expected that whatever SARS-CoV-2 was going to do, it was going to stay being SARS-CoV-2, and we'd be dealing with just that. And of course, now we know that this virus does have the capacity to mutate and throw off new variants, and we've seen that most dramatically with this most recent variant, the so-called Omicron strain. And some of that is probably just a numbers game. I mean, I think there has been about 1/3 of a billion people infected worldwide. And so even for a virus that's not very good at mutating, we're giving it 1/3 of a billion chances. So, it's kind of Russian roulette. Sooner or later, something's going to appear. So anyway, so just in ending, back to where we were, I guess, in sort of February and then early March at USC, when, like everybody else, this virus first started really getting our attention. And Peter and I, we met each other back then because we realized that at USC, we've kind of got two big things going for us. One is we've got, world-class scientists and doctors and epidemiologists and public health experts and a whole bunch of people whose day job it is to research things like these viruses. And there was this immediate, everybody coming together, jumping onto Zooms. How can we work? How can we help? What sort of research can we do? So that's, probably our, really big intellectual asset. But then the other big asset we've got, of course, is our community. 45,000 students, I think 45,000 staff, about 7,000 or 8,000 faculty. We're like a small city in California. And we're also a small city that we can collect data on, where people are reasonably compliant. Yes, no, I'd say actually very compliant, and where we have an absolute stellar student health system that really led us in how we were going to manage this. So with that, I don't know, Peter, I want to pump back to you, or we can go straight to Sarah and she can tell us about what her experience has been leading everything that happened at USC after March 2020.
Yeah, I'm gonna hand it to Sarah in a moment, but I just want to highlight this really briefly because when all of this started happening, so for me, the moment was really when the NASA imagery was published about the air pollution over China having cleared up. So I'm a recovering SARS coronavirus researcher from the 2003, from that SARS-CoV outbreak back then. Yes, 10% lethality rate. That was a frightening moment, but we dodged that bullet. When this outbreak started happening, that was that oh shit moment. When we saw those images of the air pollution having really cleared up as the simplest indication how China has really shut everything down. Of course, this is now already 6 weeks after the scientists in Wuhan had uploaded the sequence to the global databases, so that actually tests and vaccine developments were able to get kicked off at that time period. But Paula, exactly what you were saying, there was an interesting responsibility and advantage that we had at USC. And Sarah really running student health. As a faculty member, I don't think much about student health, right? This is for our students. But in fact, Sarah's responsibility was very much a public health responsibility because you actually take care of the health of the student population from many different angles, something that I would actually argue we lack in the greater society. And for us as USC, having the ability to, I'm going to say flip the switch. I know, Sarah, this was much more of a nightmare than just flipping a switch, but flipping that switch of taking student health and making your organization really taking charge of taking care of this city almost overnight was courageous. It was powerful. It was exactly what we were all about because we can do it the way we're organized. And I think now looking back, was an incredibly important step for us in that initial response measure. But with that, Sarah, I'll let you really describe more what that looked like for you over the last 2 years.
Well, thank you, Paula. Thank you, Peter, and thank you for joining it. I appreciate those kind words. So it has been a remarkable 2 years. So it's interesting. I think we're hopefully at that point in the pandemic when we talk about when our aha moment was that this was a problem, and mine was also in late January of last year of 2020, when I actually had a number of students who were arriving international students from China and from Wuhan province approach me and say, "Why is USC not taking this more seriously? Why are we not quarantining people? Why are we not testing? Why are we not acting on this?" And they reached out to me. Multiple students reached out to me. And this was before we were getting a lot of information coming nationally. There wasn't much dialogue about this. And I was like, these students know something, there's something else going on here. And then we know that things quickly evolved after that. So the response on campus has been a really fascinating challenge, opportunity to lead. I think part of what's been remarkable about it is the support from across the campus and across higher ed in general. It's interesting we mentioned a university as a small city because I think that there is very much what it is. And many of us who work in the field of college health and have had the opportunity to serve on some of our national leadership groups, we understand this public health role that we play, and we've had some experience with other kinds of communicable disease outbreaks, but nothing that's reached the magnitude of this. What's interesting, if you look at the planning that was done in higher ed, even nationally around pandemic response, a lot of it was focused on influenza pandemics. That was sort of the thing that we have been prepared for. And so early on in the response, some of that pandemic playbook came out, and some of it worked very well initially for the SARS-CoV-2 virus. Others did not hold up as well. So we think about things, the early mitigation strategies, which we put in, which includes masking, social distancing. Those are strategies that have been around forever. Those are strategies that were, at the turn of the century influenza pandemics. But those are the tools that we really had within our playbook, which is that initial approaches to kind of reduce people's chance of getting infected through masking, through social distancing, through essentially the shutdown that happened, because that's what we had. Over time, what we saw is moving more of what we think of as a catch and contain strategy. So here at USC and nationally, as we were able to have testing available, then we really moved into that strategy of, we're not only going to kind of reduce people's contact through social distancing, through masking, but now we're going to try to find cases by early testing and by implementing contact tracing. And that's what we really stood up here at Student Health over the past 2 years. We've done close to 1 million COVID tests on our faculty, staff, and students, which is a lot of COVID tests. And so this idea that we could, and this is not only happening at USC, but in our communities, by early identification of cases, getting that individual in isolation, finding out who they were in contact with, contacting those people, putting them in quarantine, that what we think of as catching and containing our cases, that was really the only tool we had then for a while as well. Beginning about a year ago, we had the promise emerge of vaccines. And as we began to vaccinate individuals, I think that really was the next chapter to where we were with COVID-19. And the idea that we hoped that by vaccinating, we could not only prevent severe disease, hospitalization, but we could actually prevent people from getting infected. And so for the last year, I think we've been in really the kind of vaccine era of COVID-19. And I'm sure we'll talk about more, and I know Paula will talk about, there's been some huge, enormous successes with vaccination. This is no longer a disease that for the vast majority of people results in severe disease, hospitalization, and death. And I can't overstate what a public health victory that's been. We know, however, the vaccines are imperfect in terms of preventing breakthrough infection. We know there may be some challenges with the duration of immunity of the vaccines. And so now I think we're really in the era of understanding how do we use the tools that are now available, the vaccine tools, where appropriate testing tools, and as this becomes something that more is what we're going to live with, and I know we'll want to spend some time talking about how it becomes endemic. But it's been very interesting. Some of the tools we've used are old, some of the tools are new. But I think one of the other things looking into the future will be is what stays behind in terms of public health infrastructure to prepare us for potentially another emerging virus like this. So maybe I'll turn to Paula to talk a little bit about vaccine, because I know there'll be lots of questions about that.
Oh, you should never ask a professor to explain something. I'll try and do it in 1 minute. I think most people probably by now are familiar with the concept that what a vaccine does is it tries to fool your body into thinking you've had an infection so that your body mounts the immune response, as we call it, which is making antibodies against the vaccine, thinking it's a virus. And then what's important is that at the end of that, once you make antibodies and you make an immune response, once you've taught your body that there is this threat out there, your body then kind of stands down a little bit, but it kind of retains a sort of a patrol in your body. There's always going to be some antibodies floating around in your blood that could stick to and neutralize any new coronavirus if it came into your body. So that's the basic idea. And we have lots of different types and platforms of vaccines that, some work better for different diseases. What I think was truly amazing and frankly extraordinarily fortuitous was that when SARS-CoV-2 first emerged, it also hit a time when we had some really significant technological advances in how we make vaccines, and technological in terms of, frankly, just speed. So again, most of you have probably heard of RNA vaccines. The Pfizer and the Moderna ones are RNA vaccines. And the amazing thing that they bring to the table is that rather than having to spend, 2 to 5 to 10 years refining and developing them, you can almost pretty much type it into a computer and have a machine that will spit out the RNA code, which when it's put into a muscle in your body, makes a copy of the little blobs that are on the outside of the coronavirus. I think it's time to bring out my puppet. So this is like a little coronavirus puppet. It's not real, don't worry. But these blobs on the outside, these red spikes, this is what we call the spike protein. And this is how the virus gets into a cell in our body. And what the vaccines do is they really make just the spikes by themselves. So having these little spikes in your body, again, fools the body into mounting an immune response against it. We will all say it, but these vaccines worked beyond, I think, everybody's wildest dreams. Sometimes people say to me, yeah, but you have to have 2 or 3, or is it going to be 4 doses, as if, perfect's the enemy of good? And I often remind people that if you want to pull out your yellow vaccine card of your children and just take a look, you'll see that many of our vaccines need multiple shots to be optimal. Now, why is that? Well, I think I'd explain it simply as the first time you get a vaccine, you get the attention of your body kind of starts to make antibodies against the vaccine decoy. But then that immunity can wane, and it can wane sometimes quite quickly. Your body is kind of like, okay, that was a one-off. We're never going to see that again. But then if you come in with a second vaccine with a boost, it's like your body goes, whoa, okay, right. This thing is, I've seen it before. It's obviously going to be around. It's obviously something I really need to pay attention to. So what happens is the level and the quality of the immune response you make just goes up, logarithmically. And so this is also what happens when you get a third boost. And I think what we've seen very convincingly with Omicron is that whereas people who've had 2 shots of the RNA vaccines are protected from serious illness and death, which is, of course, what we care about, they are actually very susceptible to getting what are called breakthrough infections, which is how we describe an infection in somebody who's previously been vaccinated. But having a third shot, having that third so-called booster, just gives a really, significantly greater level of protection. So as this disease changes and evolves, as our immunity changes and evolves over time, some of which we can't yet predict because we don't know what's going to happen in a year until we're a year from now, what we may see happening is tweaking of our vaccines. Yes, there may be in the future a recommendation that you should get a 4th dose. There may be a recommendation you should get an annual booster, combine it with your flu shot. You also get a COVID shot. The type of COVID vaccine may change. At the moment, everybody's getting their little red spikes that came from the original Wuhan strain of the virus. And we know that, especially with Omicron, that's actually very different. It's got 50 changes in its spike protein than the original Wuhan strain. So maybe we'll have a booster that looks more like Omicron than the original strain. All these things are possible. All these things can be done quite easily and simply because of the technology of RNA vaccines. And I, for one, just sometimes wake up in the middle of the night and I think, Thank goodness SARS-CoV-2 emerged in 2019 like it did. If it had emerged 10 years previously, we'd be in a lot more trouble.
So thank you, Paula. That's actually, I think this is an interesting segue to where have we come from, how we have had the advantage of being able at USC to really pull together science, technology, together with medicine and care, to really get us to a place that we look back and we're like, all right, that wasn't bad. We did okay. But we now take this snapshot in time and we now need to start looking forward. And we need to start, again, if we look back, we're like, I think that everybody would agree that most of what has happened in this viral evolution is pretty much mainstream. There is no huge surprises that have happened amongst a number of different possibilities, right? It's not fully predictable because there's always multiple possibilities, but those that did happen all made sense. And before I actually go forward, I also want to say really quickly, for those of you who are submitting questions, thank you. We will accumulate them for the next sort of few more minutes, and then start answering them to the best that we can. But I wanted the three of us to now start looking forward a little bit, and I know that's uncomfortable, right? Because we can't really quite predict it. But Sarah, you mentioned something important, right? And that was like, hey, we'll see what sticks. Also in terms of how do we actually work together with the population around us, right? We have built new technologies like health check, okay? Something that we had to throw together to figure out, okay, how do we monitor symptoms? And it was a gate to get onto campus or not getting onto campus. But those kinds of things are also powerful tools to actually understand how do these respiratory diseases emerge, right? There's this question around what is the next variant? What is that going to look like? So how do we deal with these next set of outbreaks? How do we evaluate which one is worrisome or which one is going to be more mild, right? This very ongoing debate about Omicron right now. So there is the virus, there is the disease, there is maybe the public health framework, and then there is this topic of long haul, the post-acute COVID-19 syndrome, which affects a small fraction of people who have been infected, but a small fraction of 70 or 80 million infected Americans, even in this country alone, that's a big number. We need to be very thoughtful about how do we deal with that. So who wants to take a first shot at any one of those topics that today and forward?
So that's a lot of topics, Peter. So I'll take a stab at it, and maybe I'll take a stab by adding another one, which is the real impact of the pandemic on the non-COVID things. And by that, I think about its impact on mental health, its impact on health related to delayed healthcare. And so some of what's driving us, we certainly have a lot of pressure to say, let's get back to normal, let's get back to doing these things. We also have to think about, and part of that is that return to normalcy that we think is that will help address some of the educational deficits we're seeing, particularly in younger children, address some of the mental health challenges we're seeing really for everyone in the pandemic. So I think it's important to remember those things as well as we think about controlling the virus. What are the other impacts of either the virus itself or some of the things that had to be put in place to control it, which are really going to be challenges. As we think, and I think underlying all of this, and I'm looking at one of the questions in the chat, is in many ways the pandemic and the response to it has fractured our society a little bit in terms of belief in things. We have a part of people who sort of agree with, understand what Paula was saying, which is like, these vaccines were incredible, right? And that's my view. It is incredible how quickly we were able to produce a vaccine that's this effective. Many of the ways in which the virus behaved, as Peter mentioned, are really predictable, right? We didn't know what direction it would take, but it did kind of what we thought it would. And yet that uncertainty about dealing with the novel virus has also created a certain segment of our population where it's created a lot of distrust in the vaccines, distrust in the constantly changing kind of conditions that we've been experiencing for the last 2 years. So I don't have the answer to that, but I think really looking at where did the health communications go right in the pandemic, and where has it kind of gone off track, and created groups of individuals who aren't able to partake in the vaccines or choose not to partake because they're not able to get the right kinds of information to make those decisions for themselves, who are kind of opposing some of these, what we believe as scientists are really common sense public health measures. And so that's part of, I think, what we have to learn from this. I know that's not an answer to your questions, but I think it's very important, because again, I think we've actually kind of have two groups of people who are emerging on the other side of the pandemic. We did a great job, or we really didn't know what we were doing. And I think that's going to create challenges for public health policy moving forward.
Yeah, and I'm going to I'm going to hand it over to Paula in a moment to talk a little bit more about what she expects to happen in terms of the viral evolution. But to comment briefly on, Sarah, what you were saying, I think there's an interesting opportunity for us as we go forward. I think public health as a conversation, something that affects each one of us individually. It's not something, oh, society, that's this other thing. I might choose to participate or not choose to participate. I think everybody has become much more aware that society is us, it's all of us. And part of that is, how do we deal with disease, with potential disease, right? So how do we stay healthy? A lot of these conversations have been quite fascinating, actually, and I think actually a lot of people have come along on that journey, all the way with this uncomfortable bit of science, right, where we might learn something today that we didn't know yesterday, but with what we learned today, we actually have to update what we said yesterday. We're not just changing our mind, but it's actually evidence-based, because we're doing science every single day. We're learning something new. We're putting it back into context of what we knew before. And we're trying to figure out, does it impact the way we interpret the current situation? And this process of scientific progress is important for all of us to wrap our head around. And I think there is a pretty big part of the overall community that is actually coming along on that journey. And that might in fact speak to a really, I want to say, a bright future, right? A future where we take public health much more seriously, we take our own health much more seriously, we're taking staying healthy much more seriously, mentally as well as physically, right? The total package. I think we will see a lot more, and this is maybe a little bit of my own bias, right? Because I'm very much focused on, can we diagnose disease early? Can we intervene early? Can we stay healthy as we grow older? And I do think there's an important opportunity as we go forward. But Sarah, you're absolutely right. We have to take the entire population into consideration there. But Paula, do you want to comment a little bit on what are the various possibilities? I know it's going to be a spectrum. What are the likely possibilities in terms of that viral evolution as we go forward?
Sure. So I think, maybe I'll frame that as, a year from now, where do I think we'll be? And, people often talk about this idea of pandemic versus endemic. I think we all recognize that COVID ain't going away permanently. It's not going to be smallpox. So we're going to be stuck with it forever, and it'll become, hopefully, very similar to the other 3 coronaviruses we currently have that are endemic, that just cause kind of nasty colds, and most people are exposed to them as children, but we never hear about them because they're kind of no big deal. The difference between endemic and pandemic, I think I'd frame as, when a virus is endemic, it's kind of predictable. You know what's going to happen. Maybe it'll be a virus that every winter we anticipate we'll get a little COVID uptick, in the same way that we get, flu season. We'll also have COVID season. And we can further manage what that looks like by vaccination. I think one of the really, if I had to encapsulate the value of vaccination in one little sentence, I would say that what vaccination does is it makes COVID infection predictable. We know that you are not going to end up in, dead and in the ICU. You are going to have either no infection, or if you do get infected, you're going to have either, no or relatively mild kind of, at the extreme, a bad cold. So that's where I hope we're going to be, that enough people understand this and get vaccinated and continue to maintain their immunity if we find in the future that that requires annual or whatever time interval boosters, that people understand this is not a failure of the vaccines. This is not scientists lying to you or changing the story. This is just, as time goes on and we monitor these things, there'll be, current and updated good advice. So I hope we can get to that endemic situation where everybody has enough immunity that this COVID-22 or COVID-23 is kind of no big deal. I will just temper that by saying, at the same time that this may become endemic in the United States, it could remain pandemic in other countries where there are low vaccination rates. And so there's always going to be the opportunity for this virus to evolve, because it's just a numbers game. The more people who are infected, the more chances it could evolve. And so it could throw off another variant like Omicron that is markedly different than anything we've seen before. And so our prior natural immunity, if we've been infected, or our vaccination, doesn't prevent us getting breakthrough infections, which then just kind of keeps everything going. But again, if we see that, the great thing about our vaccines is we'll be able to pivot and make something that's better adapted if that turns out to be necessary.
Paula, there's this interesting question, because I think there was quite a bit of consensus of what this evolution is going to look like. And when Omicron emerged, there was this like, oh, this could be that next version that, hey, if we're lucky, this might play itself out as much faster spread, which has to do with lower lethality as well. Typically, they go hand in hand, right? This is not like a movie where the virus becomes more lethal and spreads faster. Those two things are actually quite difficult to happen simultaneously. But at the same time, of course, as we were sort of attaching to that hopeful glimmer, we forgot the fact that we were still in the middle of a Delta surge. So a lot of questions around when do we know where we are as we go forward, and what are the right responses around that? Any additional comments?
I think it comes down to that if we have, I guess people have called it herd immunity, which ideally we'll get to through vaccination, because I wouldn't wish COVID on anybody, even a very mild one, because you are, it's Russian roulette. You don't know if you're going to be the person who has a bad outcome. But if we get to high levels of immunity in the population, then again, things become predictable, because we know that even if new variants arise, we already have such a strong foundation of immunity that a lot of people won't get infected. And it's much easier to top up an immunity, if you like, than to sort of start from zero. So again, I think the way to making this endemic, the way to making this something you don't think about more than one day a year when you go for your annual physical, and it's like, have you had your flu shot? Have you had your COVID shot? If that's the only time you ever think about it, then humanity's won this battle, and that's what I hope we get a year from now.
Sarah, I was hoping that you might comment a bit on long COVID. And as you're thinking about that question, I'm going to just touch on one topic briefly, and that is really are the other interventions. So both the drug development that has happened in parallel, in particular with the Pfizer drug now being approved, which is specifically an anti-SARS-CoV-2 drug, as well as other symptomatic treatments. We have had an ongoing trial coming out of USC here to really treat the disease from a symptomatic perspective. And the idea being that if you can push the symptoms down, you give the patient more time, you give the body more time to actually respond and overcome the virus. And those combined measures are likely going to be, again, and we are still in the middle of that trial, of course, but these combined measures are likely important as we go forward, because as we go forward, newer versions will arise where a current specific drug might not work anymore, but the symptomatic treatment will again come back into play to really ask that initial response before we have the more refined versions available. Again, that is a whole other discussion that we should have. But Sarah, maybe a little bit on long COVID. What is it that you have seen, and how do we wrap our head around it, with the diagnostic challenge around it, with this multi-organ issue around it? Any thoughts on that?
Yeah, so really interesting set of questions. So we definitely are seeing, and I think what's challenging about long COVID now is separating people who may have symptoms related to the infection pre-vaccine and people who were infected after the advent of vaccinations. So we definitely are seeing, I think we focus a lot on the hospitalizations, we focus a lot on the people who end up in the ICU, but we absolutely are seeing, significant numbers of individuals. And I think we're focusing on the people who have, the level of symptoms that are disabling to them, whether that's respiratory symptoms, cognitive symptoms, neurologic symptoms. And these are all things that we shouldn't be surprised about with a viral infection, right? These are things that make sense from a biological standpoint. We also know that there's probably a group of individuals who are not being diagnosed with long COVID, but particularly young, healthier individuals who may have very mild symptoms. They may have experienced loss of taste or smell, may have had a period of slight neurologic or cognitive effects associated with that we may actually be under the radar. It does appear that individuals who get COVID-19 right now who are fully vaccinated have much, and particularly up to date on their vaccinations, have much lower symptom levels and are much less likely to develop both the longer term symptoms or even the shorter term symptoms. So again, when we think about the role of vaccination, that's another piece of the role of vaccination. The other thing I'll say is that even in individuals we're seeing who are young, healthy, and a lot gets made of younger, healthy individuals not being at risk, people who are unvaccinated or who aren't up to date on their vaccinations have pretty severe disease. They're sick, they're debilitated, they're off work for several days. They have symptoms. They have, in some cases, mild pericarditis or mild respiratory illnesses or mild neurologic symptoms. So even the Omicron variant in an unvaccinated individual causes a disease that has real significant health consequences. So I think we're going to have to see where long COVID goes, particularly what we see with long COVID in individuals who are up to date on vaccinations, and whether that becomes if that is significant. But I think we do believe the vaccines are protective. But I'll make a comment about some of the antivirals and the other sort of things we have. So I think it's important that we're developing those tools. We certainly know that, for example, the first sort of antiviral treatments we had were the monoclonal antibodies, which are very variant specific. So many of them are no longer useful. We now have some specific targeted antivirals, which do appear to be effective in preventing hospitalization and death. I would say where we are now, though, with both drug development and treatment development is this isn't going to be a virus, I think, that we're going to have an antiviral for that's going to make it go away, and that's going to become the treatment of choice. We have antivirals that are being developed that can help mitigate some of the severest consequences, but I don't see them right now as being at a point where they become a cornerstone of our efforts. The cornerstone of our efforts really are still going to be in control of spread through early identification of cases and testing, and vaccination. So I don't know if Paula has any other thoughts on the antivirals.
No. I completely agree with you. You know, antivirals are not going to take the place of vaccines. A similar analogy might be, you know, we have anti-flu drugs, Tamiflu, things like that. I'm not going to pass up on my flu vaccine just so I can get flu and then within 48 hours have to get a telehealth appointment and then get out to a pharmacy and get a drug and take it when the virus has already been given quite a significant toehold in my body. So these drugs that are coming along, like I think it's called Paxlovid, the one from Pfizer, look amazing. But currently, you need to receive them within a couple of days of getting symptoms. So there's just logistics around that. We don't have a lot in the States at the moment. And, poor Pfizer, they're ramping up to make these drugs. And there's also one from Merck. But, these are complex chemistries to do this. And, no matter what the order that the Biden administration has put in for the drugs, it's going to take months and months before we can get these. So please don't go expecting that you can, just kind of carry on life as normal and not get vaccinated. And then if you wake up with a sore throat one day, you're going to call your doctor and they're going to FedEx the Pfizer drug to you. Please don't rely on that. Instead, by having everybody, and, Sarah, you said something and I thought that's a good way of saying it. You said up to date on your vaccinations. Like in the same way we think about vaccinations against any disease, we understand this concept of being up to date. Let's all be up to date on our COVID vaccines based on our current, best information. And then that's the way to make this virus endemic, to make the outcome to you, if you get it, entirely predictable, and frankly, to make it go away so we can all get back to our lives.
Yeah, I think it's, and actually the Tamiflu example was actually a really good one, because over the past while now, the way even that was used was actually quite interesting, because certainly we now know that for the flu, if you and your family are all vaccinated, your likelihood of severe disease is substantially reduced. But of course, it's not 100%. And in fact, if anybody in your family or in your immediate surrounding gets infected, or if it's in your family, I should say, because that's the only evidence I have. But again, this approach is very sensible. Your family physician might actually prescribe Tamiflu for the entire family to protect the rest of the family from getting severe or getting real disease, right? Because again, you intervene very early on. It's this additional part of the overall safety net, right? Vaccines is going to be the major cornerstone for all of this. Testing, I think, is going to be another huge part of us understanding where infection hotspots start popping up. It's the participations with masking, social distancing, etc., and the drugs all at the same time. So there's this fabric that we're building, and I think it comes back to a little bit to that communication again, right? How do we bring our total society along, that it's not just one silver bullet, it's a fabric, it's a safety fabric that we are building. Within that safety fabric, you can have a huge amount of fun. It can all be very enjoyable. That forward-looking normal is actually a future normal that's really, really fun. We might, in fact, not every winter or every spring be out of business for a week or two with a respiratory illness. For those of us teaching in classrooms, for example, right? I mean, this is, when I look back, I'm like, every spring semester, it was like, well, okay, when is it going to hit me? So if we can get as a society to a point where this can play itself out in this way, that again could be a very, very good way of moving forward. I wanted to point briefly to the power of testing, and Sarah, you mentioned that before, and Paula, you mentioned as well that our technologies today, thankfully this happened in 2019 and 2020 and not a decade earlier. With our routine testing today and monitoring outbreaks and the ability to very rapidly sequence at really large scales, 1 million tests at USC, I don't know how many of them, but probably a significant number of them were sequenced as well, so that we have to actually the full genome of that virus. And we can actually, when Paula just threw over the fence being like, oh, there's 50 mutations in that virus. I mean, that conversation 10 years ago would have been like, amazing, right? Today, well, we do this and we can actually do this at scale. We can, in fact, test at scale, and we can at scale identify emerging variants as well, and then figure out what does it mean to upgrade a variant to a variant of interest. You know, when does a variant get a Greek alphabet, a Greek letter, you know, attached to it? Any additional comments? And maybe, I don't know whether one of you wants to comment on the Deltacron aspect as well, and what might have, yeah. I knew that the virologist is going to be like, yeah, that's fine.
All right. And then maybe we should take some, I'm just seeing lots of questions in the chat. Yeah, Deltacron, which sounds really scary. It sounds like it's a recombination, as we say, a mix of Delta and Omicron. And the idea was if somebody gets infected with both simultaneously, the virus gets confused and mixes itself up and creates this you know, awful Deltacron. It turns out that that was an artifact of a lab that does a lot of what we call the sequencing. They got some kind of cross-contamination, so that's really been debunked now. So it was great clickbait for a couple of days, but Deltacron doesn't exist. Doesn't mean the virus couldn't mutate or do things. This mix and matching is what influenza does all the time, but so far this doesn't seem to be in the sort of repartee of the coronavirus.
And again, I think, Paula, what you're mentioning here is really super important, right? That as a global scientific community, right? Because we have labs out, there are labs out all over the world, right? Everybody is running as fast as possible. Mistakes they will happen inadvertently. They absolutely will, but there is a lot of checks and balances where immediate follow-on work is showing up, be like, all right, let's double check, let's figure out, let's do the evidence build. Okay, this is all very standard scientific process, with the big difference being like that we are doing this in the context of public health. Okay, I understand that's a little nerve-wracking, but that's okay, because at this point we are actually good enough and we have the technologies to really power an approach like that. Sarah?
Yeah, no, I think the surveillance, the testing's been amazing. And I mentioned, you know, we were doing 35,000 tests a week at USC, and it was widely reported that the second case of Omicron in Los Angeles was a USC student. And I don't think that actually was the second case of Omicron in Los Angeles. I think it was because we were actually, the way we were doing the testing on campus, we had the ability to identify somebody who had a virus that appeared to have the genetic pattern of kind of missing proteins that we knew were associated with Omicron, and then immediately get that sequenced within less than 24 hours. We had that sequenced, which is really remarkable if you think about it. You know, I think the idea of surveillance for communicable disease is a longstanding practice within public health, right? And by surveillance, we're always kind of watching, we're looking for the emergence of outbreaks. But what we're really talking about is genetic surveillance in a way that we really haven't been able to do before, right? We're not only looking for a case of measles or a case of mumps, but we're actually looking for, you know, we've been doing this some with influenza where there's some genotyping being done to look for emerging variants, but now definitely with Omicron to actually look for, with COVID, to look for those emergence of genetic surveillance. So can we have early identification of strains or variants that might, one, require a different vaccine approach or require a different, put that strain at risk for resistance to antivirals. So, and I think we're going to be able to do it when we think about what are some of the positive parts, or what are we going to take away as a scientific and medical community post-pandemic. This may be actually one of those things that we're now able to do, which will have huge value, I think, for other infectious diseases.
Yeah, a super important point. And it's actually, in fact, technologically and scientifically, we are actually able at scale to not just test for SARS-CoV-2 and its variants, but test for the influenza viruses, the rhinoviruses, like pretty much the full spectrum of respiratory illnesses causing viruses in a single test. So this is there. We have to get to a place where this is scalable at a cost that make sense for a society. But again, I think that is that next step that's going to be, because it's just going to be commonplace as we're going forward. And with that, we should be able to really identify these outbreaks, characterize them with confidence, and then respond accordingly. For the last couple of minutes, I know there were a lot of questions. There were a lot of questions around long COVID, and just as a there's this bit, of course, like, why does long COVID happen with SARS-CoV-2 infections? And there are a couple of things that are really different about this virus than what we have seen in some other cases. But I'm going to put a caveat to that in a moment. But one thing that has been really distinct is that the virus is causing a multi-organ disease pattern, right? It's not just in the upper respiratory tract, it's not just in the lungs, but it's upper respiratory tract, it's the lungs, it's the central nervous system, like loss of smell, loss of taste, this brain fog concept, the entire cardiovascular system, right? The damage that it causes in the lungs and really has a real impact on our blood vessels. In fact, in the early variants, it was the impact on the lungs was so massive that the immune system would actually overreact. It's called the cytokine release syndrome. Now, when that happens, that is again something that the body doesn't easily overcome. That actually takes time. But again, the damage to the cardiovascular system, to the heart, to the GI tract, that happens during the active disease itself. And then what lingers for a sometimes very long period of time is still something that is where the science is just kicking off, really. There are groups around the world that are trying to wrap their head around it. It's complicated because we don't have a test for long COVID that does not exist yet. There are, of course, a couple of research programs at USC, at around Los Angeles, that are driving that forward. But that is, it's complicated. There are the observations. This is from a group that we just talked to in London, in the UK, and a series of observations and fairly decently powered studies where they have shown that patients with long COVID, they can do a 5-minute walk or a 5-minute run, but then their muscles are not able to reoxygenate. They're just powered down and out for the next 10 days. And so there is a disease pattern that we really don't understand yet. But this is all going to be active research, active science. Again, we are getting a much better understanding how viral infections can really be triggers and drivers of other diseases, whether that is in the autoimmune space or, of course, now with long COVID as we're going forward. Any, Sarah, Paula, any sort of like parting comments on that topic?
I think I just have one quick one, that this is so reminiscent of things like chronic fatigue syndrome, where we understand there's probably a virological assault or something else initially that then leads to this. So long COVID is not so unusual. It's certainly in the spectrum of other things, other post-viral syndromes that we've seen. I, for one, am kind of encouraged that because this is all happening in real time, because we're seeing it, there's going to be a lot of research and a lot of information about this. And I really hope that in addition to figuring out how to fix long COVID, we also make some real inroads into fixing chronic fatigue syndrome and other similar syndromes as well.
Yeah, and I would just echo that. I agree. I think it makes sense with what we've seen with other viruses, and I think from my perspective with the population I take care of, I think we also need to have a better understanding of the more subtle aspects of long COVID, where and understanding the impact it might be having on at a low level on potentially large parts of the population. So I think the takeaway from that is, we don't want people to get COVID. If they get it, we only want them to have breakthrough infections if they're up to date with their vaccines. So I think even with the Omicron variant, the potential impact on people who are infected is real. And so I think we have to have caution with the idea that it's okay to get COVID now, which we are hearing some places.
Yeah, so again, with that in closing for those last few seconds, First of all, of course, thank you, Sarah and Paula. I loved it. I really hope that our audience, I'm pretty sure it was useful for everybody. But again, we are a research university as well, right? And I think what you saw today is also this interplay between medicine, virology, and science that can really get us to a better and better place as we're going into the future, right? We can construct our own future. We can invent our own future if we work together and allow for that interplay to actually take place. So with that, thank you both very much. Thank you to USC and the Dornsife team for setting this up, and wishing everybody a good afternoon from here on. Take care.